questions, and just feel free to stop me at any time.
Okay.
All right, so yeah, this is part 1 of 2. So first I wanted to, instead of just talking about the software, it would be nice to introduce all the concepts around this, just assuming that not everyone is familiar with MVA in tof-sims and orbisims. So before, I just wanted to list here what we expect to achieve today. So I want to transfer some basic MVA concepts in the context of SIMS data. And then I want to show how to prepare SIMS data, specifically SIMS data for MVA, and mention the most common pitfalls related to this, and also how to visualize MVA results, for the best results you have. And then I divide this into 4 parts. So these are slides I got from a few lectures I gave. So some of you have seen these slides before, some of the slides are new. So the first bit will just be an introduction with flashing some applications of what we can do with MVA. Then I will try to describe the most common MVA methods, and then talk about data pre and post processing, and then just finalize with introducing the software that will be used for the next session. Right, so SIMS data, as you all know, especially in these modern instruments, are getting every year more complex, because we can see inorganic samples, organic samples, biological samples, and hybrid samples with all these things. So back in the days, we used to only look at inorganic things with SIMS and with low mass residue, but now we can see all kinds of samples. And also it's getting larger. So now there's, for example, a typical 3D data set of that profile. We have more than 1,000 levels with more than 200,000 pixels per level and more than 1,000 ions at each pixel. And when it comes to orbisims, it's even more than 1,000. It can be tens of thousands and etc. So what we end up with, if each pixel for each level, it's what we call, it's a voxel. You have more than 2 million voxels. So because of this, multivariate data analysis methods have become popular within the SIMS community. So this is sort of outdated, goes on only up to 2017, but you can see this is a trend of publications involving surface analysis and MVA. So you can see there's a trend, it's increasing, people are using it more and more. And this peak here in 2008 is a special issue in the Surface Interface Analysis Journal. So I think it's a good reference if you're interested. There's several papers there, it's more than 10 years old now, but it's really good reference because many of the standards for MVA of tof-sims were set at that time. And I think soon we have another special issue, to update on all these things. So a few examples of what we can do with MVA. So we can use it to classify data, for example. So, when you have several spectra of samples that may be similar or not, and you want to differentiate them using the full range of your data, you can use it. So for example, here we have 6 kinds of decarboxylic acids, and we can use MVA to get the signature in the SIMS data and then separate them. So and then if you get a mixture of these 2 acids or one unknown acid, when you measure, you can use this MVA model to know which acid it is based on your classification model. You can also use it to find trends in depth profile. I feel like lots of people are doing depth profile nowadays, especially with the orbisims. So you get tons of peaks in your depth profiling signal, and you can use MVA to group them and find patterns and find trends of peaks that will be together. So that's quite useful. You can also use it to look at imaging data sets, so you can find patterns that will be related to common chemistry. And also by doing this, you can join these peaks that will have this similar chemistry, and you increase contrast. So sometimes you have really noisy data when it comes to imaging, and MVA is quite good for this, apart from the whole advantage of, figure out what's the chemistry on your surface. And also we can join all these things and process those large complex 3D data sets and identify complex chemical systems. So here's an example. You have 2 polymers, one encapsulating the other, and after MVA we can see all the peaks related to these polymers. And it's not as simple in SIMS because, as you know, sometimes the same polymer you yield 2 polymers will yield the same peak, So when you do this, you take into account the ratio between these peaks, and you can separate them, even though they have very similar signature in terms of their spectra. So this is just as an introduction. Obviously, we can cover more in detail during this these slides. So the most popular methods for SIMS data are principal component analysis, PCA, and non-negative matrix factorization, which is also known as multivariate curve resolution. So these are statistical methods. So in tof-sims, variables are secondary ions, to say in terms of statistics, and spectra are observations. And these, when you say spectra, it could be just individual spectra of samples, or they could be pixels in a map, or voxels in a 3D depth profile, or levels in a depth profile. And then nowadays we have this whole machine learning field, and these methods, they will fit in this subcategory called dimensionality reduction, which is part of the unsupervised learning. So we don't input anything into the data, we just process it and get the patterns out of it by reducing the dimensionality. So both PCA and NMF will be in this category. So let's now take a step back and try to understand at least the very basic concepts of PCA and NMF. So there are several ways to talk about PCA and NMF, and specifically for PCA, I think a really good way to talk about it is think about the geometrical relations that it involves. So if we consider a tof-sim spectrum, with several peaks, but then to simplify it, we just go and pick only two of these peaks. So imagine that this is a very simple spectrum with only two peaks. And to make it more relatable, let's say these are two peaks from PDMS. Everyone working with SIMS have measured PDMS before, I'm sure. So, and then we take various measurements of these two peak spectra. So measurement number 1, and then we have intensity for the peak at 28 will be I1, and for the peak at 72 will be I2. And then you go on to the second measurement, and these intensities will vary, and you keep going, right? So if we get these intensities that we recorded and plot them against each other, you may get something like this, right? The more of I1, the more of I2, which is expected because they're both coming from the same molecule from PDMS. So if you do many more measurements, you may end up with some line here showing this correlation between these two peaks. So what PCA does is simply finding the direction of what we call the highest variance. So in which direction the data is changing the most, in simple terms. And then it just rotates this axis that we set with I1 and I2, so that the principal component number 1 will point in this direction. And that's why we call it dimensionality reduction, because before, in order to explain, let's say, the quantity of PDMS, we had to, here we have I1 and I2, but they're both redundant, as we say. Like, if you know how much of I1 is there, you also know that there will be a lot of I2. So if we change this to a new axis, which is the new principal component, you can only say, well, there's this much of PC1, and PC1 represents PDMS. So that's the basic concept of PCA. It's axis rotation when you think about the geometry. And then if we change this and not plot PC1 against PC2, so it's just changing this diagram, just rotating this diagram, you see that for PC2, the points are randomly distributed around the 0, which means it's just noise or variation. In terms of SIMS, it will be noise most of the times. So in this case, we only need PC1 to talk about this data. So the terms you see in the literature is one of them is the scores, and scores is basically the new coordinates in this new set of axis. So a high score means that the point is high up in the PC1 axis, and you will see here for PC2 it's just randomly around 0. And PC1, it just increases. So it means like here you have a lot of PDMS and here you don't have a lot of PDMS. So obviously, when you do this, you lose the whole structure of the whole chemistry of your data. You know you have PC1, but you don't know what PC1 is. What we know it's some contribution from 28 and 73, but it's just PC1. So that's why we also use what's called the loadings, and these are projections of, this PC1 here onto the original axis. So you can see it depends on how much you rotate. You see the contribution to PC1 from both the original axis. So again, PC2 will show one point into each direction, which is indicative that in this case, that's just the noise. And PC1 shows almost equal contributions of 28 and 73. So 0.6 and 0.7. So sort of is almost like a 45 degrees if you still want to talk about the geometry. So it's a 45 degrees rotation. So these are the loading. So equal contributions of 28 and 73 for PC1. And that's when you make the interpretation. Well, I know these two peaks come from PDMS, so PC1 must be representing the amount of PDMS in the samples. And in this case, PC2 relates to error and noise. So this went down from 2 variables to 1 variable. So it's a 50% reduction. Well, you see, obviously, in this case, it may seem useless, but it will be quite useful when we do several variables, several thousands of peaks. So before we continue, just a few examples. These are from a lecture I gave. So it was supposed to be interactive, so I wanted people to kind of guess where the principal components are in each of these examples. But it will be difficult via Teams, right? So I will just show you where the PCs are for each of those. So here is the example we just saw, PC1, PC2. Remember, it's a rotation. So when you set PC1, PC2 has to be orthogonal. It has to be in this direction. So we always start by setting PC1, and then you work out PC2. So in this case here, it's PC1 is in this direction. So when you get the projections, you see there's more contribution of M1, which it was in the old example, it was the 28, right? So M1 contributes more to PC1 than M2. So, and this is reflected in the loadings. There's a different ratio between them, and it's related to how much it rotated. And if you observe, PC2 is always showing the same. Now, this one here, the angle is higher now, which means this is a higher contribution from M2. So it's the opposite case. So like the 73 in this case contributes more. So this won't be 72 and 28 because, in PDMS, we know the contributions may be similar. And the final example will be this one, where you have this cloud of data far from the origin. So if we apply the same rule, we just rotate the axis, PC1 will be here and PC2 there. And then it looks like you have equal contributions of M1 and M2. But that's not true. If you look at how much this varies, it's not like the first case. And that's why we always have to do something that's called mean centering before we do PCA. So what we do is we move the origin of your set of axes, so I1, I2, to the average point of your data. So we just move it there, and then we find the principal components. So now you see that PC1 is actually higher contributions from M1 than from M2, not equal contributions. And that's very important, because if you don't do this, you have wrong interpretations. But every time, if you don't do this, always your PC1 will actually show the average of your data. So the average of your data, it's just M1, M2 and equal contributions. That's it. So if you have more peaks and don't mean center, you're always going to get your average spectrum for PC1, which is not really informative. You don't need to do PCA to find out the average. So it's important to do mean centering. So one final example here is now the more of I1, the less of I2. So there's an anti-correlation, not a correlation. And again, if we just do PCA without mean centering, look at that. The loadings are just the average again. So when we do mean center, now PC1 will show M1 against M2. So that's quite important. Like when they point at different directions, doesn't mean it's wrong. It doesn't mean it's noise like we were seeing before. it can also mean that M1 and M2 are anti-correlated. So on the positive side, you're going to have all the peaks that are likely from the same compound, and the negative side from the peaks from a different compound. And there are ways to figure out whether or not this is noise. When you do PCA, you get how much of the variance was captured by each principal component, and they're always ordered in these terms. So PC1 will always show real variance. So in this case here, you know it's actually information about your data, not noise. And then obviously in SIMS, we don't only have 2 peaks, we have more, but we can only use this geometrical example up to 3 variables because that's the maximum number of dimensions we can see, right? So again, that's quite difficult to see, but what you end up here is you find out PC1, and then PC2, and PC2 will be sequentially orthogonal to PC1. And then you can get projections like here to see where PC1, 1, 2, and 3 are. That's, this starts to get a bit more complicated. It's not as intuitive as the 2 dimensions. And that's the maximum we can go. So we have to make an abstraction now and think about several variables. So yeah, that's just what the loadings will look like for PC1, 2, and 3. So 1 and 2 in this case will be from one compound and 3 from another. As you see, they both anti-correlate with M3, but they correlate between themselves. So before we talk about more variables, just a more realistic situation, still in the 2 peaks case, we tend to do repeats of all measurements, and then these repeats will be around some mean value if they're coming from the same sort of sample. And when you do PCA in your scores plots, you have these clusters grouping your samples. So that's why in several papers you see these sort of plots grouping the peaks, grouping the observations or the measurements with different colors. So if we do then PCA for several mass peaks, based on what we know about the loadings in 2 dimensions, when we do hundreds of dimensions, they will look like spectra because it's just a series of peaks with specific ratios among themselves and pointing upward and downwards. So it's quite helpful because when we see this sort of structure, it reminds us of spectra, so we can identify patterns that come from a knowledge of the chemistry. And it's quite common when you have several variables to represent scores with scatter plots. So you plot PC1 against PC2, similar to what we did for I1 and I2 original variables. Now we do with the principal components, and that's quite useful to group sets of observations. So here you can see the advantage. Like we have thousands of peaks, so we could do scatter plots of everyone against each other. But if we do PCA first and then find all the peaks that are related, we end up with just a single scatter plot that, as written here, is from thousands of possible scatter plots with original variables. PCA enables the whole data set to be shown in a single graph. So with the right interpretation, you can explain your whole data with a single plot instead of trying to find the patterns one by one. So that's the introduction about PCA. I will talk about NMF now. It's difficult to know whether it's too fast or making sense. So if I will just stop for like 5 seconds, see if anyone's got a question. Okay, we'll continue. So the other one is NMF. So to talk about NMF, it's the geometry example is not too useful. So I like to think about the interpretation you get out of NMF in the end. So basically, we have this assumption that a surface consists of a mixture of molecules and materials, or materials. So I'm representing each material by one color here, right? Yellow, red, and blue. And then when you do surface analysis on this sample, you get mixed signals, so like XPS SIMS or any other technique. So you're gonna get a spectrum that's a mixture of all the molecules, like it's yielding peaks from all the molecules on your surface. So what we do with NMF is we try, based on the statistics, we try to unmix this signal into pure signals of these components. So basically, what we try to achieve is separate the contributions of these pure molecules that will be yielding specific peaks at specific ratios. So basically we get something that looks very much like the spectra of the original, of the pure compounds. And also we also get the concentration distribution of these things if you have several variables, because now you know the individual contributions, the individual pure signals, you can get the concentration of each for a sample, specific sample. So as a comparison, PCA, as I say, will capture the variance, but NMF will unmix your signal. And note it's notice that it's non-negative, so it's easier to interpret. There are several pitfalls around this, but in a way it's easier because you don't have all these negative contributions. So it's all about, okay, if you make the interpretation right, you separate this into compounds and you know how much of it you got for each of these samples. So here, like F1, you have a lot of F1 for the oranges, but not a lot of F2, and the opposite for the purples. So moving forward, we can think about this, applying this to images, because as I said, images is where you have loads of pixels and variables and observations. So we just use the exact same concept, but now we use each pixel as a sample. So we can do all those same scatter plots. But actually we have the advantage of knowing the relationship between these observations. So like the spatial relationship. So you know how to map these things because they have original coordinates in X and Y. So we can do PCA, NMF, and then see them instead of using scatter plots, we can see maps of the results. So to try to make things a bit less complex, I created this synthetic data set using photographs. So the idea is if we could photograph 3 subjects at the same time, but then per pixel we're not detecting light, we're detecting secondary ions. So I made this data set with some real SIMS data, and if you get the total ion intensity of the total spectrum, that's what it looks like. And then you can see here the total image clearly looks like a mixture of three things. You can see the doodle door and also myself. So if we do PCA across all these pixels, as I say, it will capture the variance. So you'll find pixels. So it's color-coded here, like purple for positive, green for negative. It will find pixels that show similar behavior, that correlate. So that's what you get. And you may find things that you couldn't see in the total ion image because the signal is too weak. But as a comparison, if we do NMF, as I say, it will unmix the signal. So in this case, it will actually separate them into the original images. So, and with the spectra related to those original images. So both have given you the same information. It just depends on how you interpret and what you want to get out of this. In this case, it's quite easy to see what's going on with NMF because it's just separating these 3 images into individual ones. And you can see here in the original one, we couldn't see this cat. It's my wife's cat. We could only see the dodo and myself. So sometimes you have very weak signals that can only come out if you do this sort of unsupervised analysis. Sometimes we just skip it because it's just too weak to be detected. And it's worth pointing out that I added some noise, and as a consequence, the final results will still show some mixing. You see here that you can see the ear of the cat in the dodo door. I don't know if you can see here the dodo door in my living room, which is not true. So yes, it's due to noise. And as I said, we can use it to find hidden patterns. But look at PCA, it also was there. It depends on how you interpret things. So these are the original images. I thought it was worth showing this photograph I took at the door, especially at this time, because we can't go out. And also the cat, gorgeous cat. So we talked about images, but we can use the exact same concept for depth profiles. In this case here, like we have this organic inorganic interface in a depth profile. And as I said before, each depth level will be a sample. right? So if we do exactly the same for this depth profile and the same sort of comparison, you see that PCA, you show PC1 going from a positive value and swapping to a negative. So you can think of the loadings, obviously looking at the chemistry of the peaks, that positive are related to the organic peaks at the beginning of the depth profile. The negative is related to inorganic peaks towards the end of the depth profile. And then PC2 is quite interesting because it goes to positive only at the interface region. So the peaks related to PC2 are actually peaks present only at the interface, the organic and inorganic interface. And negative will be the no-interface peaks. That's one interpretation you can have. But you can get the same information out by doing NMF in this case. So you're going to, instead of 2 principal components, you have 3 NMF end members, as we call them. So the red one will contain your organic peaks, the yellow one, your inorganic peaks, and the blue one, the interface peaks. So both techniques showing the same information. And obviously NMF in this case is easier again to interpret because it looks like a depth profile. You know, you don't have negative things. It's like only positive things like we have in chemistry.
Sorry to interrupt. I have a question.
Sure. Yeah, sure.
How did you got from having the 2 components in the PCA to 3 components in the NMF?
So obviously we do them separately. They're not related. So that's one thing about NMF, and I didn't give this detail, but you have to say how many components are there because it's an approximation. So you have to say. So in PCA, we'll find as many principal components as original variables because it's a rotation. In NMF, you have to say specifically. So in this specific case, the way I got to this point was I did PCA first and identified 2 different regions with different chemistry. So the inorganic, the organic, and the interface. And then I did NMF and I said to the algorithm to find 3 components. So that's a common question, like how to define how many end members are there. And it's quite common to use PCA first before you do NMF, because as NMF is easier to explain and to visualize, interpret. But you have this downside of having to input how many components are there. So if we use PCA first, it may help us to figure out how many we use for NMF. Does that make sense?
Yeah, this makes sense. Thanks.
Right. So it relates to the next 2 slides, actually, which I don't want to go into all the details of that, but just a little bit of the mathematics behind this. So for both methods, we can organize our data in a matrix that we call A with all the intensities of the peaks in columns and the observational spectra in rows. So what we do is matrix algebra. We factorize this matrix into 2 matrices with smaller dimensions in this case. So in one of them will contain the loadings, the other one will contain the scores. And then you have constraints in this factorization. For example, in PCA, we know it's a rotation, so we need to keep this orthogonality between things. So I'm leaving a reference here, which is my PhD thesis, if you really want to learn about the mathematics of this. But for both techniques, it's exactly the same kind of factorization. So it's a 2D matrix into 2 other matrix. So they're both part of something called factor analysis, because we're factorizing a matrix. So with NMF, again, what you get out of the factorization is a matrix with concentrations and another one with the pure spectra. So you can see it's a linear combination of concentration times spectra. So I don't know, like 1% of blue, which is all those peaks together, plus 90% of the red. So that's how you separate it, and it's how it relates to the mathematics behind this. And again, related to the question, that's how you set it. That's why you have to set the number of components, because this factorization, we have different columns for each of these columns for S and rows for L, which are representative of the number of compounds or end members you have. So here is, in this example, you have only 2. But we can have 3, you know. So you have a third one, a third set of peaks, and a third set of concentrations, or 4 or 5, and so on. So that's the disadvantage of NMF. There's not a single solution. So for us to achieve this, it's an approximation. So you have to use an algorithm to find the approximate solution. And also, you can find solutions with as many compounds as you want, because it's an approximation. So it's not exact, and it needs some backing up of the idea. So that's why PCA helps a lot sometimes to identify the chemistry first. And also, to get to this matrix, you may think that, well, with imaging or 3D, it's more than 2-dimensional. So what we do is called unfolding. So as we're treating each pixel as a singular observation, we just stack their spectra or the pick lists on top of each other. So that's called the unfolding. And then you have exactly the same matrix we had in the previous 2 slides. So you can just do the factorization, you can do PCA or NMF. But what we do is, once you find these concentrations, because it's an imaging data set, you don't have to just look at the concentrations. You can put them back together in the mapping configuration, because you know their coordinates, and then you get this kind of visualization. So these maps are the concentrations of the end member from NMF, but now mapping into intensity scale and with their correct XY positions. But that's how we get from that matrix factorization to this visual representation of the results. So yeah, to make things a bit more realistic, that's why the sort of things you get. Like this is kind of an interface. This is a polymer blend on the left and a substrate on the right. So it's a cross section. And that's what you get. You get components for the interface, for the polymers, or for the substrate. Okay, so that covers the 2 main methods, at least the basics. We can always discuss more later, or if you have questions, or I refer you to the PhD thesis, my PhD thesis, which is the first chapter. It kind of covers all these things in a bit more detail. So I want to talk about data pre and post processing, because we need to prepare the data before we do these things, otherwise we get wrong results. And also, once we get the results, what to do in order to visualize them. So that's what I want to talk about now. So the first thing is something called variable selection. So when you're doing SIMS, each channel in a detector can be treated independently, and that's considered a variable. So we could just get each of the channels in a spectrum and then use them as variables. And this is quite often done with data such as XPS, ADX, or Raman, because you only have a few hundreds of channels, or a few thousands, and that's feasible. You can just do it. But for SIMS, tof-sims, and even more for orbi-sims, for a single unit mass, you have thousands of channels. So like here, several channels. So when you expand this to the full spectrum, you're going to have millions of channels. So you have a huge matrix to deal with. And it's just time consuming, memory consuming. And so it's quite difficult. So what people do, and what we in the SIMS community do, is we pick variables before we do any MVA. So the most common strategies for doing this is peak picking. Most of you must be familiar with this. We just select regions in the spectrum where we will add together all the counts. So we integrate these peaks and then turn that into variables. So here, from all these channels, we're making 6 variables, each of the variables related to 1 peak. We can also do organic, inorganic binning. So up to, I don't know, 200 mass units. Most of the times, it's not for every kind of sample, but things on the left-hand side of the nominal mass will be inorganic, and the right-hand side will be organic. So you can do this kind of separation. You can get all the counts from the left-hand side and right-hand side, and then turn them into variables. So it's kind of compressing the data. So in this case here, from these thousands of channels, you end up with 4 variables, which may be all right because you only had 6 before, and maybe these ones are very tiny. So maybe you may get away with this. So it's quicker. It's not too different. Or you can do nominal mass binning, which is just for each nominal mass, you get all the counts there. So in this case here, from the thousands of channels, you go down to 2 variables. So that will be mass 1, mass 2, right? If this is 55 and 56, that will be 55, 56. So just doing the same for that example I showed with 1, 2, 3, 4, 5, 6, 7 different peaks for the same unit mass. Again, if we pick, we get 7. And this is tough data, right? You're doing Orbi data nowadays, and you know that there'll be many more. Sometimes inside this single one here, you're gonna have 3. Sometimes you have isotopes, and, you know, so it's there will be many more than 7 if it's Orbi, for sure. If we do the inorganic binning, now we have only 2 because it's only left-hand side and right-hand side. So we're losing information, right? We're going from 7 to 2. So that's quite dangerous because you're adding together things with carbon, nitrogen, oxygen that may have different spatial distributions, for example, if you're doing a mapping of different concentrations of different samples. So you have to be aware of this. If you're doing binning, you're compressing the data and losing information. And with the nominal mass binning, in this case, all these peaks become a single variable. So that's the danger. So you have to consider sometimes, you know, it's a simple system, and for unit mass, you only have a single peak. So you can do this. It's just a matter of knowing your data. And these are the most common strategies. There's a few others. There's some new ones in the literature nowadays, but these are the most common ones. For any case, what you end up with is a list of peaks, right? In this case here, 1 for each unit mass, and here several more for each unit mass. Another thing we have to worry about, I won't give much detail, it's something called Poisson correction, but that works only for TOF. So these slides are from a lecture I gave before the Orbi times. So variables, they have different amounts of noise or error. So what we do, and the statistics of these microchannel plate detectors are described by the Poisson probability distribution, so the counting statistics of these channels. So we can approximate the noise by the square root of the counts. So what we do with Poisson scaling is roughly divide each of these areas by the square root of them. And by doing that, we normalize the level of noise. So for the noisier variables, we give a higher weight. So like here, the higher masses of your spectrum, you have higher intensity after the scaling. So you kind of make them, try to normalize them to the level of noise. Because if you don't do this, sometimes you do PCA, for example, and your principal component will show just differences in noise. And this has nothing to do with your chemistry. So you'll be getting information that's not meaningful. You'll be just showing you which peaks have more noise. And these are usually the low intensity peaks. So we'll group, for example, low intensity peaks against high intensity peaks. And that's not useful. So we do this to correct for the noise. Again, it only works for TOF. For Orbi, when you do Orbi, you see that there's not much noise there, but the way Orbi the peaks are detected is different. It's not based on counting statistics. Use the Orbi trap detector, and I'm not really sure the statistics behind this. And hopefully, in the future, you know, we're gonna get papers and people will start figuring this out to think about what's the best correction for Orbi if we need one at all. But Poisson correction is only useful and only appropriate for TOF data. Yeah, so lower signal to noise ratio get enhanced. Another thing is the normalization. So in broad terms, we normalize things to account for differences in acquisition conditions. So for example, acquisition time, you know, if you acquire one sample for 10 minutes or and the other one for 5 minutes, obviously the overall counts of the first one will be higher so you want to correct for this. So we normalize by the total counts. Or if there's any change in the primary ion beam current, so higher current, you more peaks per second, so you have to compensate for this. Or secondary ion yield affected by charging and topography effects, which is quite important. So in all these cases, if you don't normalize, your MVA, your PCA or NMF will show just differences in regions that are related to topography or maybe just acquisition times. You know, if you have 5 samples acquired for 5 minutes and 5 for 10 minutes, they will be grouped together in your PCA results. But that doesn't mean they have similar chemistries. It's just because they had similar acquisition times. So when you normalize, you correct for this. So with regards to topography in imaging data sets, this happens because, you know, there's an optimal height for detection in SIMS, and there's this depth of field. So there's a specific height where you lose signal or it just gets weaker. So this is reflected in your total ion count. So here in this case, you have regions with really poor counts because it's out of the optimal height for detection. So if you get the counts there and normalize by the total, you try to flatten this, right? You correct. So here, for example, for the potassium signal, the intention is to flatten it, right? There's all these contrast here is to do with topography, and you don't want to find this out with your MVA. You want to find out about the chemistry. So you try to flatten. But this is this is a real example, and it's not perfect several times. And that's because sometimes it's just too noisy. And when you do it, you enhance the noise. So yeah, so it's called the edge artifacts. It's due to low signal. So every time you're dividing by something with noise, you kind of adding this noise to it. So you have to be careful with that as well. And that goes back to your acquisition. You know, if you want to measure a very topographical sample, you have to have these things in mind.
Sorry to interrupt. There's a question from Akmal regarding the Poisson correction.
Okay.
Do you only do it for the NMF and not for the PCA? And can you do it prior to the PCA to overcome the issues associated with the noises?
So you do it for both, but only for TOF data. You do it for PCA and NMF, but you only do it for TOF data, not ORBI data, because in both methods, both PCA and NMF, you'll be looking for the statistics of these intensities, and you need to correct these intensities to normalize the noise. So it works for both. So sorry, what was the second part of the question?
The second part was, if you do it for both, can we do Poisson correction prior to PCA to overcome the issues associated with noises?
No, it's you never do it after, right? But you do it for both, and it's always before you do the MVA method, always. And also before normalization. So it's the first thing you do.
Okay, thanks.
Also normalization, like I know many of you are dealing with large area imaging with the stage raster. And when you do this sort of things, it's quite likely that you're going to get differences because there's a tilt in the sample or things change because it's a long acquisition. So it's quite important to normalize things when you're dealing with large area imaging. So it corrects for what we call this patch artifact. So here you can clearly see the patches. So the stage moved and there are quite different patches here, like 1, 2, 3, 4, 5, 6. And when you normalize, you get rid of that. So this will not be picked up when you do MVA. Again, the downside is for this specific ion, the counts were really low here on the right-hand side, and you get this enhancement of the noise when you do normalization. So you have to be careful. Also for depth profiles, it's quite important to normalize because sometimes due to roughness, you get this kind of wavy patterns. So these are like roughness artifacts. And if you don't normalize, you may get a principal component or NMF and member showing this waviness as if it was some variation in chemistry. Many times it's just a variation due to topography. So when you do this, you flatten the profiles as well.
There's a question from Matteo.
Okay.
Do you normalize by the total ion for the whole image or normalize by total ion per pixel?
It's per pixel. Each spectrum, imagine you're doing several measurements, not an imaging. So each pixel is and then you make them into pixels. Each pixel its own measure.
I think you've muted your mic, Gustavo.
Oh, it says someone muted me. Sorry. You didn't get the answer, right? Are you? Can you hear me now?
Now we can hear you again.
Okay, so so what was the question? Sorry, I don't know if I.
The question was if you do normalize by the total ion for the whole image or normalize by total ion per pixel.
Oh yeah, so it's per pixel. I don't know if you heard it, but as I was saying, each pixel is an individual measurement, and then if you imagine that these measurements are different samples, they will have different acquisition times or topography or things like this. What you have to do is divide by the total intensity of this individual measurement. So it has to be per pixel. So to simplify is that you get the total ion intensity image. It's usually the first one you get in the Surface Labs software. Just divide all the other images by this one. So divide per pixel.
Okay, thank you.
So this one is only for PCA, the mean centering. So as I explained in the two variables case, right? If we don't, we have to move the origin to the average of your data. If you don't do this, the principal component 1 will show you the average of your data. So here, an example using an imaging data set. If you don't mean center, that's what you get in the first principal component. And look at the scores. The scores are just the average of your data. So it's not really informative. When you do mean center, you get real chemistry. So you have this inner part here in red to these ions, and the blue red to the other ions. So it's really important to mean center when you're doing PCA. And you don't do it for NMF because NMF is non-negative. And as you can see here, whenever you mean center, you bring in lots of the data points to the negative side of things. So they will be all ignored for NMF analysis. Then these are not mandatory things, these last two things, but they're quite useful. So still in the pre-processing, there's something called the pixel subsampling, which is it's only useful for very large data sets like imaging or 3D data sets. What we do is we only use part of the pixels to do NMF and PCA, and then we apply these results to all the pixels. And you can see here the difference in time. Like if I do all pixels for a specific data set against 1% of the pixels, so it's much quicker. And it's backed up with literature that several times you don't need all the pixels in tof-sims data to find out the principal components, because neighboring pixels are correlated, not due to the spread of the ion beam, but also physically correlated. It's to do with the phenomenon happening there. So it's quite useful because it saves time. So yeah, that's what you get NMF with all pixels and then 1% of the pixels. Quite similar, allows you to have the same conclusions and saves you time. Other one, pre-processing, it's what we call stitching, or in more technical terms, is matrix augmentation. So the idea is to join maps, or in the same way when you do several measurements of several samples, you join them in a single matrix. You can do several images, and then not just treat each image as a single measurement. You can just treat them all together, all the pixels in a huge final matrix together. This is to save time and also to constrain your solutions, right? Because if you do PCA and NMF for each data set, you may get different results. If you process them all together, you're going to have one consistent set of results. And that's quite useful, especially when the samples are related, So in this case here, like there's a paper with more details, but what we did was we had 3 cross sections or 4 cross sections. So we mapped these cross sections and then joined everything in a single matrix, and also added measurements of the pure materials that we expected to be in these cross sections. And that helps a lot, you know, because even though it's unsupervised, with some way helping the analysis by giving pure signals there. So this will kind of, it's quite likely that if you add something that's is very homogeneous and pure, that one of the end members will be there, because, you know, all pixels here, they have the same chemistry. So it's quite strong in terms of the variance. So it will help. So in this case here, the practical terms, what you get is kind of a color code that relates to the chemistry. So you don't even need the spectra in this case, because you know these are your standard references. So you just see, okay, this green reference is present here, where it's green. You know, it's quite useful to this sort of stitching. So it's good for saving time, it's good for having a consistent set of results as well. So these are all the pre-processing. So I would just talk a little bit about the post-processing, things you do after you get PCA and NMF. And these are all related to visualization, because, you know, what you get is a matrix with the numbers, and there are good ways to show these results. Even though the dimensionality is reduced, you still have to show it in a way that's easy to explain. So there's something called the biplots, which is showing scores and loadings together. So as I said, you can plot scores against each other, right, in PCA. That's already quite useful, because you can see here, like, these are the carboxylic acids I mentioned at the beginning. They were grouped together in this diagram, so it shows you what they are. But then you can also project the loadings, the original peaks, onto this diagram. So it's kind of showing, it is showing the loadings together with the scores. And then you can see here the most characteristic peaks for each of these groups. So that's quite useful. Not every time, sometimes it gets a bit messy, but depending on your size and finding the right threshold for which ones to show, it can be quite useful, you know, to convey information, because now you can see here, like, these are related to the tricarboxylic acid, these are the adipic acid, and etc. You know, you already have everything in one diagram, and you can do it in 3D as well. You can add a third principal component here and do exactly the same. But then if you're not able to rotate it or have a video, things like this, it may be confusing for some people. So for imaging in PCA, it is quite useful to use first diverging color maps, because we have things going from 0 to positive and 0 to negative. So instead of having those classic color maps where you go from black to red or from white to some color, you have this, that this 0 is black, and then positive values go to a color, negative values will go to another color. And we can also do it for the loadings. So it's quite easy to match one to the other, right? Anything blue here matches the blue in your map, and red matches the red. And you have the intensity distributions, like the more kind of white red or light red, the more you have of these ions. So it's good for identification, it's good for presentations, papers, and all these things. So I consider this post-processing, because you need to change the color schemes and etc. But it's just visualization, really. If you do NMF, what's quite useful as well is to overlay the intensity maps. Like, you know, in this case here, there's NMF end members with 1, 2, 3, 4, 5 end members. Each of them have their own distribution. So here I'm showing them with those classic color maps, because now it's down negative, so it makes sense to use the same color scheme as we get from the Surface Lab software. So it's going like from black to orange, yellow. But you can also assign each of these end members a color and then overlay them. So for example, you can overlay, so this intensity will be mapped into the opacity or the transparency of its corresponding color, right? So that's what you get in the end. You get this composite image with more than 1, 2, more than 5. You can even do more. You know, it gets confusing if you have too many, but that's the idea. You get all the information in one single image. I find it quite useful. It's similar to doing the overlays. You do it in Surface Lab, but you have much better contrast because you're taking all the peaks into account, and sometimes you have more confidence to say, well, this is that polymer, because they're not only mapping this ion, mapping this component, this end member, containing all these ions that are related and quite likely to be coming from this polymer. Yeah, so like the blue here is like a chromate layer on this metal substrate, just an example. For 3D, there's something you can do it before and after as well, which is correcting for topography. Because if your depth profile is something that's got topography, like in this example here, when you get the SIMS data, that's what it's going to look like, because the levels are not going to differentiate whether or not there was topography in the beginning. So you get this kind of inverted result. So what you do is correct to look like the one to the left. So here, like using this copper grades, copper and aluminium, what you get is the real distribution of copper in 3D after you correct. But you can do this after you do NMF as well, because or PCA, you just do it based on the components you get. And still with regards to 3D, the outputs are fully tomographic and also multivariate. So you have this full 3D distribution and with and all the chemical information. So like here in this slide, I have all the peaks related to these two polymers. It's the first example I showed at the beginning. But at the same time, it's a simple image, a video just showing the 3D distribution. So it's quite useful. And again, here I'm doing an overlay, but the 3D version of an overlay, right? I'm assigning a color for each. Then I'm doing some the transparency is arbitrary here, just more for the purple. But each of them have colors, and the way to define where you will draw the figure is based on an intensity that you set. And then we can do these isosurfaces. So when we do the practical thing with SIMS, and we can show you how to do this. But it's quite useful to show your 3D data with this kind of rendering. So you can show different levels. It's more technical, but sometimes to convey the idea or show a nice slide, it's good to have this 3D rendering of your results. And again, if you do it for the original ones, sometimes it's too noisy, it won't look as good. When you do after NMF, you're joining peaks together, so you're enhancing the signal to noise, and things will look much smoother and much better. Okay, so I don't know if, oh, sorry, yeah, lots of, those are the last few slides. Okay, we'll finish very soon. Just wanted to introduce SIMS MVA in preparation for the next one. So before, I will just give 5 seconds again for questions. Right, so SIMS MVA, it's a MATLAB-based app for doing this sort of MVA analysis of any kind of really analytical chemistry data. But the focus is on tof-sims, but you can use different kinds of data sets there. It was developed throughout my PhD, and the main motivation was to make it quick and accessible to everyone in the research group. So all things I was doing was based on the problems we had in the labs. So it was always in collaboration with some other colleagues. So it's got all these visualization tools I mentioned and some others. And you can do PCA, NMF, and also something called k-mean clustering for different data structures. You can press the spectra, images, depth profiles, 3D data sets, and to get these results I showed here. So there's a website, mvatools.com, where you can download, or you can always write to me if you have questions on how to download, how to install. I recommend you using the MATLAB version. So if you have MATLAB, it's much easier to run SIMS MVA, but there is also a version where you just run it standalone. There's also a paper explaining some details about the software. So I just want to show this timeline. It's, yeah, I mean, it was from the lecture. I will show you as well. I started in June 2014 with just, some functions to process, to do MVA, or to do PCA actually of some spectra I had from some polymers. And then I decided to name it PCA GFT at some point. And I decided to make a software. So it was called GFT, is just my initials. So it's only in 17, that's when I changed it to SIMS MVA. And and presented it to actually people outside of University of Surrey. And that's when I started to, give it copies to other people, presented at conferences. And then when I finished my PhD, I had 17 users. And now there's more than 100 registered users. I don't know if everyone's using it every day. Some people just use it once, but it's quite nice to have people everywhere using it. So for the next time, what I'm hoping to do is have a practical session with some examples using Surface Lab to export the data. So to show you if you don't know how to export it, and then SimsMVA to process and visualize, and hoping to do some of the advanced ones, such as the stitching or the 3D, some very complex ones to perhaps inspire you to do it as well. Okay, so that's everything. Sorry if it took too long.